Hello, everyone, and welcome to today's webinar, Biosecurity for Genome Engineering, Safety First, and the SeqScreen Solution for Functional Predictions. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Inscripta. To learn more, visit them at inscripta.com. Now, we want to encourage you to participate today by submitting as many questions as you may want during this presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have any trouble seeing or hearing this presentation. Now, this webinar is educational and offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain those credits. I'd now like to welcome our speakers today. First, Dr. Beth Vitalis, a biosecurity specialist at Inscripta. Joining her is Dr. Krista Ternus, a bioinformatics specialist at Signature Science. And finally, Dr. Todd Treangen, an assistant professor of computer science at the Department of Bioengineering at Rice University. Beth, Krista, and Todd, welcome. You may now begin your presentation.
All right, well, thank you for this opportunity today. I want to thank LabRoots, our collaborators at Signature Science and Rice University, my Inscripta colleagues, and everyone for their interest. So today we will address biosecurity for genome engineering, goals and challenges, and the power of working together. And I'm happy to be tag teaming here with Krista Ternus and Todd Treangen, who will present a remarkable resource, SeqScreen, that is providing a solution for functional evaluation of engineered or otherwise novel sequences. So what we hope you learn today, first, why biosecurity is important for everyone's safety and to safeguard advanced bioengineering technologies, such as microbial genome engineering. Second, we will explain what is needed for proactive biosecurity screening practices to identify and help prevent biorisk. And as an example approach, outline Inscripta's multi-pronged screening for our Onyx microbe genome engineering platform. Third, we will discuss challenges to predict biorisk in engineered organisms. Then Krista and Todd will describe a computational screening platform that is user-friendly, open source available, and provides robust biological characterization of input sequences. Finally, we will see SeqScreen prediction come alive with a few real-world applications. So to start, we recognize that genome engineering capabilities are empowering scientists to explore biology at extraordinary scale. And now we can make discoveries and drive impactful innovation that will help improve human health, promote and expand sustainable materials and fuels, agriculture and food supply, improve medical care, and help meet other pressing needs. So there's tremendous promise and excitement in microbial engineering. However, as access and scale of technology expands, we must pay attention and work together to respect and protect the power of all bioengineering technologies. Okay, so biosecurity protects everyone. Well, what do we mean by biosecurity? So I could condense into a definition here. Biosecurity systems include procedures intended to protect humans, animals, plants, and all organisms from being harmed by biological agents. And these bioagents that may be a safety threat are commonly microbes or toxins. We have lists of known biorisk-causing bugs and proteins, but we also must consider novel sources, such as SARS-CoV-2. For successful biosecurity, some questions we need to address: how can biorisk be introduced? Where might it be coming from? And how do we identify potential biorisk before it is a problem? What does it look like? So science is moving fast and biosecurity must keep pace. All right, so this first question, how might biorisk be introduced into synthetic biology technology? Well, there's two major ways. It may be deliberate, an individual or entity with nefarious intent, that bioterror character that we all fear. The other major source is inadvertent, an accidental event. Famous examples of such unforeseen results in previous biological research have been fatal to mice. So we are calling this inadvertent source bioerror. It arises since biology is complex, and the outcomes of biological or bioengineering technologies may be difficult to predict. And also synthetic biology technologies are advancing, scaling up and rapidly diffusing to broad populations, increasing access. So biorisk may result from either mischief on the left or mistake. So think for a moment, which do you think is a growing concern for the microbial genome engineering? Well, while we need procedures to block malicious use, for advanced high throughput technologies such as democratized genome engineering, bioerror is becoming a focal point. All right, so let's go to that second question. How do we recognize biorisk? It is important for all researchers, get my slides to catch up with me, to assess safety in their own work. And here I highlight a landmark study by a National Academy of Science committee in 2018. They laid out an off-cited framework for evaluating biorisk potential in new synthetic biology technologies, including microbial genome engineering. And I want to note that this risk evaluation framework was leveraged by an engineering biology research consortium sponsored and DHS-funded malice analysis workshop series in which Inscripta participated. And we hope all industry will take action to promote safety culture. So the report highlighted drawbacks in this current list-based risk assessment. What an organism can do is more important than what it is. And on a practical level, sequence screening needs to move beyond bad microbe to sequence of concern, as we'll hear more later. All right, so now that we've established a need for biorisk prediction in microbial genome engineering, as an example, I will describe the Inscripta impetus and process for proactive sequence screening for engineering microbes. So the Inscripta Onyx platform, I'll describe in a nutshell, allows for the massive parallel engineering of microbial genomes. So to consider the number of variants that can be rapidly engineered, we scale up in 2 dimensions: the number of genome-wide targets represented on the x-axis, as well as the diverse edit types, SNPs, indels, knockouts, etc., on the y-axis. And so this huge variation, up to 10,000 edits per library, enables enormous genotypic and thus phenotypic variation. And the platform includes the instrument, the software, and reagents. It is all optimized and accessible, so customers design their own genome-wide edits. And while we are excited about the power this large-scale diversity generation brings to genome discovery, we're mindful of the potential biorisk this could introduce. So in particular, researchers are now better able to explore uncharacterized parts of genomes, and we consider complexity of genomic interactions. The potential of bioerror comes into view. And as a final note here, Inscripta is committed to safe and responsible use of this technology, and we aim to help ensure that this responsibility is held up across the rapidly advancing genome engineering industry. All right, so safety first. This customer empowerment facilitates high-impact discoveries, yet must be addressed in a biosecurity system. So here's an overview of our multi-pronged proactive approach that addresses the major ways biorisk may be introduced. So there's 3 major screening steps shown here. I'll have a few more details on following slides. So first, upfront customer screening to help prevent the known bad actors. And we trust approved customers, so the sequence screening, the second 2 prongs, are primarily to help prevent bioerror. Communication is key. Biosecurity processes involve coordination across groups. At Inscripta, it's a company-wide priority. And so our biosecurity perspectives and practices are communicated with customers, and all are welcome to learn more on our website. Okay, so first, we aim to block deliberate misuse of the technology, a rare event, yet may be devastating if it happens. We follow industry standard practices to screen against government banned lists that include individuals most likely to have nefarious intent. Trade.gov is a source. We anticipate virtually 0 true positives, and also point out that knowing your customer comes in the event of a hidden sequence screening. Follow-up protocols include dialogue with customers as needed to assess legitimate use. And although we do not anticipate misuse, customer screening is essential for everyone as technology reaches more and more people. Right, so let's look now at the computational sequence screening. It's carried out at two levels, the input genome and edit design libraries. So we have an in silico screen of customer-provided genomes as a first pass at biorisk detection, comparing against lists of known bad genes and proteins. The second in silico screen is done on all customer-directed edits of very large libraries. And for this step, we sought a functional prediction rather than a list-based approach. And that's where SeqScreen enters in, a functional biorisk prediction software, about which we will all learn more soon. And a biorisk alert on either level will invoke manual verification and, if warranted, dialogue with customers, as mentioned on the previous slide, to discuss how to move forward safely. Libraries passing the final screen will go forward to manufacture, and reagents are shipped to happy customers. Okay, so point out that SeqScreen is supported by the government agency IARPA, and you'll learn more soon. It's provided a solution for many of the encrypted sequence screening needs that we've encountered, several of which we believe are shared across the bioengineering community. The software can handle enormous plexity of our libraries. We've had 500K plex library screened in less than 2 hours, variable size sequence inputs, and it supports in-house screening, which is critical to ensure confidentiality. So SeqScreen will assign a function to novel sequence variants to help predict the biological input. And important for all industry is that the cost of a screening system is not a high barrier. To our knowledge, this SeqScreen is the only microbial functional prediction for biosecurity use cases that is open source. And descriptive values are long-standing collaboration and ongoing interactions with the SeqScreen team. Okay, so I will end on the note that security is a shared responsibility. Community gathering sparks collaboration. In late 2019, a few of us were addressing the unique considerations for biosecurity for genome engineering. And we had thoughts of, let's start getting folks together to chat and work together to begin to solve the problems we were facing. In particular, Sarah Carter, Krista Ternus, and I had some memorable visits at SynBio Beta that year, which planted the seed for such a focused gathering. And then soon, Inscripta was fortunate to partner with Megan Palmer and Catherine Brink of Stanford University to landscape and organize a virtual workshop that was held last November. So 27 participants with diverse expertise converged to initiate dialogue and collaboration on industry practices, resources, and opportunities for microbial genome engineering. The report is now published on our website, and the key outcome is a desire for inclusive, dynamic community. Let's keep the dialogue going. And with that, I will turn it over to Krista.
Thank you, Beth, for giving that introduction and mentioning SeqScreen Software. I wanted to share some more information about SeqScreen in case anyone in the audience is interested in downloading and running that locally on your systems. At the bottom of this slide is the website address where you can go and identify more information about SeqScreen. On the wiki associated with the GitLab page, we have quite a bit of documentation that'll explain how to download and run the software, as well as what the different workflows will output for you when you run SeqScreen. We have 3 different modes in general that you can use for SeqScreen. The first is called fast mode, which is our default mode. So if you don't specify what mode you'd like to run in, you're going to automatically go to fast mode, which will utilize Diamond for associating each sequence with its most likely protein ID and functional annotations. The next option is sensitive mode, which will be, as it suggests, more sensitive in its analysis, but it can also take more time because it uses BLAST rather than Diamond. But sensitive mode also comes with the additional benefit of running many other tools within our pipeline. So those are pictured here on the left in green, and you will get a number of those tools output in intermediate files. So even though the outputs may not show up in the final report, there's lots of goodies in there if you're interested in any additional information about functional annotations that you can get with sensitive mode. And the final mode is ONT mode. That's intended for analyzing long reads or contigs. With fast and sensitive mode, there's an assumption that there's only one protein coding sequence per analysis, per read, whereas with ONT mode, we assume that there are multiple open reading frames within a read. So when you look at your outputs, you'll have multiple lines of output with ONT mode, depending on how many open reading frames are present within a read, whereas with fast and sensitive, you'll have one line of output per query sequence. And we definitely encourage you to use Conda to install our software because there are a number of different packages that need to be installed for dependencies. And we also have instructions for how to run this locally as Docker or Singularity on our wiki page if you are not able to connect to the internet on your local systems. So please take a look at our documentation and try it out. We'd love to get your feedback on that. In general, the SeqScreen final output format comes in two different flavors. One is a computer-friendly parsable file that allows you to easily look at things if you're going to be plugging into the SeqScreen outputs into a downstream pipeline that you computationally want to analyze further. So that's a TSV file. The other output type is a HTML report that you can interact with. So it's a point-and-click framework. And pictured here on this slide, you can see the alignment of one of the sequences that was a query to the database hit. So lots of interesting interfaces that you can click on there and see if you want to individually look at your sequence outputs. The final thing I wanted to highlight as far as running the software is that we have added a number of different flags that you can run with our software that will modify the parameters or note what tools you'd like to run in the pipeline as optional additions. And you should do whatever works best for you. Not everyone has the same exact use case or research goal in running our software, and we've tried to include as many options as possible to make this workflow customizable for different end users. So please take a look at all the different options that we have and think about what makes the most sense for you. And Scripta is a great example of using SeqScreen because they've taken certain portions of the output and plugged that into a larger pipeline that they use for biosecurity, and that's what Beth was describing earlier. So we definitely encourage everyone to use this and customize it based on your workflows and use cases. So now moving past the logistics of how to install and run the SeqScreen software, I wanted to talk a little bit about how we put together the software, what our motivation was, and what kind of gaps we were trying to fill at a high level in developing this software. And Beth hit on this earlier. The big idea was that we wanted to move away from simply classifying a sequence as dangerous because it hit to a pathogenic species of concern. Or if you have a list of taxons of concern, and you screen your sequence against that list, it hits to a certain taxon that's flagged as dangerous. Well, that is certainly beneficial, and there's a place for that, and we're not trying to replace taxonomic analysis. We recognize that there are many open source tools available to do taxonomic classifications, whereas there are not very many open source tools available to do functional characterization of microbial sequences, especially when it comes to biosecurity applications. So that was a gap that we were trying to fill here in making sure that we were trying to develop something that was going to be of benefit to the community, and we hope that it is. If you're interested in reading more about the motivation behind the development of our software, there are a few references to papers on the right on this slide, and we encourage you to take a look at those and read them after the webinar. And I wanted to share an example just for illustrative purposes of how results would differ if you were running a traditional DNA screening protocol for biosecurity purposes against the Biological Select Agents and Toxins list, or the BSAT list, compared to running our SeqScreen software, what different outputs you would get. So pictured here in this table, the first 4 rows passed the BSAT screening protocol. In other words, these particular sequences were from proteins that were not on the BSAT list. However, when they're analyzed with SeqScreen, you can see that they have some functional potential that could be damaging to the host. Specifically, they're capable of disabling organs. whereas the last two rows of this table are sequences from proteins that are similar to the virus that causes smallpox. So they're from vaccinia, so they're not necessarily from that virus, but they have homology to it so that it does raise a flag when you're running the BSAP pipeline. And if you look at the actual function of these sequences, they aren't inherently harmful by themselves. So the first one is involved in normal viral replication processing. The second sequence is involved in the viron structure itself. So no sequences of concern were assigned by SeqScreen to these particular sequences. And again, we feel like the information provided by SeqScreen and the functional characterization provided is complementary to many of the other tools people are using right now, but it is filling a gap in giving you that functional information that can help inform your biosecurity decisions. And another thing I wanted to mention, as we were developing the software, there was a big gap that we noted in the availability of customized labeling or ontologies that could describe how microbes can cause harm in a host. So we spent a lot of time, and there is a paper linked at the bottom of this slide by Dr. Jean Godbold on our team, who helped us to develop functions of sequences of concern, which are customized labels that help describe how a microbial sequence might cause harm in a host. And when we think about computational solutions in software, having these standardized labels to help describe why something might be pathogenic is really critical. Otherwise, it's just a black box of something being assigned pathogenic with no real understanding or description of why. And so we really tried to make this understandable and useful to end users in the way that we catalog these sequences and integrated them into our software. I also wanted to mention that around the time we were developing the functions of sequences of concern and SeqScreen Software, other groups such as Johns Hopkins University Applied Physics Lab was working on something called the Pathogenesis Gene Ontology, or PathGo, which is conceptually very similar to what we were doing with FunSox. And we benefited from collaborating with them and working with them on their PathGo ontology while we were working through our FunSox customized labeling approach for SeqScreen. The PathGo is available, and it's an ontology that you can go to at GitHub at the link at the bottom of this slide if you're interested. There aren't currently any annotations associated with that ontology, but we are hopeful that the community will use this resource in the future to help annotate sequences, and please check that out. Another group that we spoke with as we were developing the functions of sequences of concern was the Gene Ontology Multi-Organism Working Group. And these are folks that have been working for a long time on ontology problems, and we benefited a lot from their expertise and insights in how to supplement some of the descriptions that we use for describing microbial pathogenesis. And Todd's going to tell you more about this in just a minute, but I wanted to mention that the FunSoc labels that we annotated internally with our subject matter experts were used for training and testing of machine learning algorithms within SeqScreen. So the experts that we have on our team read scientific literature to assign FunSocs to different sequences, and so we have experimental evidence for each of those assignments. And then machine learning was used to expand those FunSoc labels to all well-annotated proteins within UniProt. And that is the database that you would use with SeqScreen if you were to go and download that today. And one thing that I find helpful about that approach compared to other approaches is that the database that we have within SeqScreen contains very well-annotated sequences. So if you wanted to understand why something was included in our database and why it was given a pathogenic label, not only does it have a FunSoc label, but it also has quite a few gene ontology terms and other annotations within UniProt that you could go back and look at to try to understand why it was classified in one way or another. This is different than other databases like the virulence factor database or the VFDB, which does not include information about why a sequence was incorporated into their database. So there's no additional metadata or ontology terms associated with those sequences. And we feel like this is a strength of our database because that allows end users to help interpret results with more confidence. So with that, I'll wrap up and pass it over to Todd. And Todd, I trust that you are going to have fewer technical issues than me. So go for it.
Thanks, Krista. Yeah, that was, so I'm just going to continue the discussion here and going to actually do just a slightly more technical deep dive into how do we computationally detect these fun socks that were just described. And so on your screen right now, we have just a toy example of an example DNA sequence fragment. And what I'm showing here is just to convey the idea of what is SeqScreen doing? Well, when it's looking at this example DNA sequence fragment, essentially we want to identify potentially fun sock containing regions indicated in red. And for this mock example, we have cytotoxicity and degrade ECM, which are two fun socks inside of the total set. And then we have a region in the middle that is a housekeeping gene that has no fun socks assigned. And so just conceptually here, this is, again, if we're thinking of what SeqScreen is actually doing when it gets handed a collection of DNA sequence fragments, this is the task at hand. But one question might be, how does it accomplish this? So on my next slide here, this is going back to what we saw previously. It's really this human-in-loop fun sock prediction process. So we started off with the Biocreation team at Signature Science, where they performed deep literature review, reviewing several relevant scientific publications and sources of evidence for placing specific sequences and proteins into a given category of a fun sock. From that, we then developed a training data set to train our machine learning algorithm to then go and then apply that to unseen sequences and kind of iterate over this process where this is a really key part of this whole project and really required this closely knit team effort over multiple years, where we would annotate through expert biocuration, update our training set, predict, and then hand that back to the Biocreation team for review. And after doing that through multiple iterations, we ended up with a software tool that was closely vetted by the Biocreation experts, but also was built off of best practices in terms of the machine learning side as well. So I have next slide is going to just get a little bit more into the details of how did we train SeqScreen. Well, without getting into too many specific details, which is outside of the scope of today's presentation, I would just like to say that we started off with a database of about 90,000 proteins, which had high confidence annotations for training. And what information is used for training here? It's going to be gene ontology terms and keywords, among other features. And these now all have labeled with fun socks thanks to our Biocreation team. So a subset of Biocreation labeled with fun socks, and then we went out and trained and expanded this and for the iterative process I just described. So again, without getting too many details, there are two different machine learning approaches. One was where we actually performed feature selection first, and then fun sock classification, and then one where we just given all after getting rid of the rare features, we just went straight into fun sock detection. And again, the manuscripts that Krista had shared provide additional details in terms of the specific machine learning approaches that were used. But needless to say, there was both feature selection based approach and just a full set of features were used for the fun sock detection. And then again, this would tie into the iterative approach that was just described. Now, just high level, I have one figure of results here. X-axis will indicate one of the fun socks that SeqScreen detects or classifies, and on the y-axis you have kind of an accuracy value, and there's multiple colors, lots of rows, all hard to read, but the main message here that I would like to convey is there's variable performance for any specific machine learning approach for any individual FunSoc. However, if you look at the ensemble, which is where we combine multiple machine learning approaches, which is that red line, you will see that the performance overall gets much more consistent across all of the FunSocs. And these FunSocs, again, referring back to my original example, could be cytotoxicity, could be antibiotic resistance, et cetera. So this slide is really to convey that SeqScreen has a variable performance across each individual FunSoc category, and we can help mitigate this by using an ensemble machine learning approach built out by the team. And that's the main message for this slide. So now I just want to walk through two vignettes before we conclude here, and really just two quick examples. One is a near neighbor microbe detection kind of example, and vignette 2 is quote unquote a needle in a haystack example. So I'm just going to walk through both of these now. So the first example is the goal is, let's say, to identify FunSocs within a microbial clade with near neighbors. And by near neighbors, I mean a clade that contains genomes that are very similar to one another within 1 or 2% average nucleotide identity across the length of the genome. Perhaps less, perhaps slightly more than that, but more or less that's the framing of the problem. And to look at this, we ran state-of-the-art taxonomic classifiers to compare performance. And then in this example, where we have a set of very closely related genomes, in this case it is Clostridium botulinum with Clostridia sporogenes, SeqScreen was able to correctly identify FunSocs associated with the bot A toxin. So I will walk through this now, and then results slide next. And what do we have on the screen? On the top, we have a table that indicates for two isolates, the near neighbor microbe example, that Clostridium sporogenes and botulinum. Top row is CSPO, bottom row is CBOT. Any red cell will indicate the column, the method indicated in the column, incorrectly labeled it with the other genome, and anything in yellow is indicating that it correctly labeled it. And so as we look across this, we see variable. The main message here, is that you'll see variable kind of performance across the state-of-the-art taxonomic classifiers due to the closely related genomic content. So it's a very hard taxonomic classification task, given these are all very closely related, and one could. So the point of showing this to everyone is that in this example, what we really care about is, do these genomes contain a FunSoc of concern, or in this case, can we identify the bot A protein? So I said previously that SeqScreen was able to, but now what happens if we remove the true organisms from the database? What if we remove the true organisms? And if we do that, some further confusion can happen with the methods. And then what if on the SeqScreen side, we go remove all of the CBOT and the associated proteins from the databases? Turns out that SeqScreen is able to identify distant homolog and still correctly identify FunSoc containing hits inside of the genome. So this example is a very short vignette into SeqScreen's ability to characterize FunSoc containing sequences inside of near neighbor genomes, which is a very useful and important feature for biosecurity analysis. For the next and final vignette, I'm going to talk about something called the needle in a haystack. And what I mean by this is really imagine there's a sample of millions or tens of millions or hundreds of millions of sequences, and say just a handful of those contain a FunSoc or a sequence of concern here. And so we want to be able to even in those examples, we don't want to limit ourselves to only looking at things that occur above a certain frequency or a certain threshold. We want to be able to still, even in these complex mixtures or really high throughput data sets, still identify rare signal inside of that sets of thousands, tens of thousands, millions, hundreds of millions of sequences. And this is exactly what SeqScreen was able to do. So what I'll now describe is the goal, which is to find differential gene ontology terms between a control and COVID-19 patient blood metatranscriptome. So we really want to look and see, so there's a publicly available data. We want to see, can SeqScreen find any signal that would distinguish between control COVID-19 negative and a COVID-19 positive samples, so 3 of each in each category, that could indicate the presence of the virus. And so we ran an earlier version of SeqScreen just to simulate an emerging pathogen example where the SARS-CoV-2 gene was not in the database. And in this specific case, the virus and case-specific gene ontology terms were identified in 2 reads in one of the COVID-19 samples. And the gene ontology terms that were associated with the differences are listed here on the slide. And I'm not going to read them all out, but essentially they're all related to suppression or induction of virus activity, which corresponds with later we validated that correspond to SARS-CoV-2 activity. So this is just one example, again, a second example of kind of the utility of running SeqScreen in cases of either near neighbor pathogens or kind of emerging pathogens inside of with kind of a trace signature. And just to visually show this again, if we dig in further to this example, through identifying these Go terms specific to the viral sequences, and only finding 2 out of many reads, this kind of highlights the capability that SeqScreen allows you to kind of pursue, given that it is an individual sequence characterization tool. We'll look at each and every read within your sample and try to assess the Go terms in the fun socks that might be of interest with respect to functions of sequences of concern. And on the slide here on the bottom, if you pull the 2 reads out that we identified, we have and we highlight as funsock containing and of concern in the sample. They have perfect alignments to the SARS-CoV-2 genome. So just as a validation step here. With that, I would like to transition and kind of just summarize on the next slide here. So overall, this genome engineering holds great promise and also poses new biosecurity considerations. And not only with my colleagues, Krista and Beth, but with the broader team and community, we need to proactively assess functional impact of editing. Inscript has prioritized biosecurity. It has a multi-pronged screening approach, very engaged with the community. We're very fortunate to be collaborating with Inscripta on this important problem, and also have a longstanding partnership with Signature Science on this important problem. And just kind of summarizing some of the points that were made earlier, plus within my section, I just want to emphasize that SeqScreen provides an open source solution to several genome engineering sequence screening considerations. And as was mentioned previously, this is not meant to be replacing any tool. This is identifying and filling a need in a use case that was previously unfilled. We obviously appreciate all of the great work that's been done in many of the areas we touched on, especially with respect to taxonomic classification. So, but the challenges remain. Genomic or original context is not accounted for, and that's something that that would help improve things more. We need relevant test sets of engineered sequences. We need improved biorisk relevant annotation, and scale up. Continuing to making sure that this can scale up is very important. So with that, I would just kind of issue a kind of a rallying call here for everyone interested. Let's work together on this important problem. And then what I mean by that is resource building and sharing, crowdsourcing, and collaborative problem solving. I think there's a lot of potential here. There's a lot of room for collaborative efforts on this exciting yet and very important problem. And again, just want to emphasize how great it's been to work with both Inscripta and Signature Science on this problem. With that, I would like to conclude just by acknowledging the great team that has enabled all of the methods that were presented today. You know, none of this happens without a long-standing and multi-year collaborative effort with Signature Science. We had a very just vast team. I would like to shout out to members in my group specifically, Advait Balaji, PhD student in my group, as well as postdoctoral scientist Dr. Leo Elworth, who have contributed to previous versions, but also the funding sources and the end users and the broader microbial research and genome engineering communities. We have received so much valuable feedback and important collaborations that have helped improve this open source solution, addressing this important need. And we truly look forward to continuing to working and engaging with all of you in the community. And again, in terms of funding source, just want to acknowledge the support that's been provided by our IARPA on the Fund GCAT program over many years. And with that, I would like to conclude and thanks again to Krista and Beth for setting up my section nicely, and also thanks everyone for attending today, and happy to take any questions.
And thank you, Todd and Beth and Krista, for that informative presentation. We will now start the live Q&A portion of this webinar. And if you have any questions you want to ask, please do so now. Just click on that Ask a Question box located at the far left of your screen, and we'll answer as many questions as we have time for. So it looks like we already have some great questions coming in from our audience. Our first question is, SeqScreen is a reference database dependent approach. How does SeqScreen behave if the DNA sequence being screened is a distant hit or no hit to sequences in its DB?
I can take that one. That's a great question. So in the example I covered in my section, it's important to note that we took all of the Bot A toxins and removed them all from the database. And so just to exactly try to get into that question, we removed all Bot A toxins from the database, and SeqScreen was still able to identify a distant homolog and then characterize that as FUNSOC containing. So that answers part of the problem, part of the question, sorry. Kind of the second half of that question, we view as future work de novo characterization approach for sequences that are just very distant and have never been seen before. So again, that we view that as future work, and happy to engage with others as we move forward and kind of look additionally into that area of kind of de novo characterization of very divergent, previously unobserved sequences.
Thank you so much. Our next question, is sequence screening against the database done on the nucleotide or amino acid sequence level?
So I will take that, and that's a great question. To maximize sensitivity of the screening at the genome level, it's both nucleotide and protein against the respective databases. And at the edit design library step, through SeqScreen, it's 6 translation frames are all sent through at the protein level. And this facilitates prediction of the biological significance, as we heard from Krista and Todd.
Thank you so much. Now, how does SeqScreen work in relation to the IGSC harmonized screening protocol?
Yeah, I can take a shot at that one, and others can chime in too. Thank you for that question. So the IGSC harmonized screening protocol focuses a lot on the list-based approach for screening, as Beth and others have mentioned today. And that has to do with looking at BSAT agents and making sure that the sequences that people are going to synthesize don't match to anything on that particular list. I think SeqScreen goes above and beyond that list-based approach by looking at the actual functional potential of different sequences. And certainly we wouldn't want to replace all of the great work that has been done in that protocol, but we think that these types of functional approaches could be complementary and could help enhance some of the insights that companies can get when they're screening sequences by looking at functional potential.
Thank you so much. And I want to thank our audience for these great questions coming in. Our next one, how does Inscripta prevent misuse of the Onyx benchtop biofoundry?
Yeah, so I'll address that. So that's the 3 levels of screening, the customer screening up front, and then the sequence screening also can address that. We also have controls within the instrument so to prevent unauthorized use. So it's all inherent within the system.
Thank you so much. Now, is SeqScreen's machine learning approach supervised or unsupervised learning?
I could hop in and take that one. It is a supervised human-in-the-loop machine learning approach, and this is kind of a key aspect of this multi-year effort. Future work, well, we have discussed exploring unsupervised machine learning approaches again to get to kind of expand capabilities out, but as is, it's a supervised machine learning approach.
Thank you so much. Our next audience member says, I'm very interested in contributing to the SeqScreen code base. How can I get involved?
Krista, do you? I can take that one.
Sure. Yeah, I can take that one. So I definitely encourage you to look at some of the links that we had in our presentation today to where you can find our open source code. And we encourage you to download and run our software. We also have an issues page on our GitLab repository, and you can post any questions you have there or feature requests that you might have in mind. But we're very happy to collaborate with anyone who is interested. So please do reach out to us through our software page or to us directly, and we'd be happy to talk with you more about how we could collaborate and get you plugged in and involved in either SeqScreen software or the larger genome engineering biosecurity community.
I guess just to add on that, I just want to emphasize the fact that it is on GitLab and open source. Just emphasize feature requests, pull requests, comments, engage as engagement would be great.
Thank you so much, Krista and Todd. We have time for a few more questions. Our next question, can interested individuals become involved in the ongoing genome engineering community biosecurity dialogue and maybe participate in future discussions?
Yeah, I'll take that, and absolutely input and participation is welcome. So a step could be to contact one of the event organizers of the workshop report or reach out to any of us. Yeah, we're happy for input from the entire community. Thank you.
Thank you so much, Beth. Our next question, where are the list of bad genes and proteins you use for genome screening coming from?
I will address that also, and this extends from Chris's description of the SeqScreen in relation to the IGSC protocol. So all of Inscripta's protocols, we've been also following and building upon the Harmonized Protocol guidelines, and they follow HHS guidelines that were put out a few years ago for DNA synthesis providers. So, and we leverage them and then extend upon them, curating available databases, and our databases continue to evolve to meet screening needs. So yeah, that was a good question.
Thank you so much. What is the status of funding for SeqScreen software development?
Yeah, I can take that one. So we are currently funded through August of 2022, and that funding is primarily to help to optimize SeqScreen analysis of long reads. So that's with the O&T mode that we mentioned today. But we are actively interested in other funding sources and other collaborations. So if you have an interest in continuing to develop this software with us, please do reach out. We would love to talk to you more.
Beth, Krista, Todd, I want to thank you for your presentation today. Would you like to provide any closing remarks before we go?
I don't think so, other than thank you all for listening to us today and participating, and we hope to hear from you and engage with you either on the GitLab page or in other ways in the future.
Well, I want to thank you, all three of you, again for your time today and for your important research. And I also want to thank LabRoots and our sponsor Inscripta for underwriting today's educational webcast. And before we go, I want to remind our audience and thank them for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone, and bye-bye.